and to Sophion for the invitation to speak today. So as David introduced, my name is Tony Lewis. I'm from the University of Portsmouth. So this is the Solent. The university is based over here, so I get a lovely view of the Solent. So I'm here to talk to you today about fungal potassium channels. So the reason why I have an interest in fungal potassium channels is due to the fact that fungal infections account for a major part of health problems across the world. So fungal infections kill more people than malaria and tuberculosis combined. Generally they affect the immunosuppressed population, and generally treatment's quite complicated because of the evolutionary conserved biology between mammalian and fungal cells. So there really is a requirement to locate and identify fungal specific targets for therapy. The most common opportunistic fungal infections in humans are through the species of Candida, Aspergillus and Cryptococcus. So there are 3 known families of potassium channels, certainly in animals and plants. So we have the voltage-gated channels here. So the subunits consist of 6 transmembrane domains, a single semi-hydrophobic pore loop here, and a voltage sensor at S4, which confers voltage sensitivity to the function. The inward rectifier channels have a structure of 2 transmembrane domains in a single pore loop, and they're intrinsically openly rectifying, but they're blocked by intracellular polyamines and magnesium in a voltage-dependent way, creating this inward rectifier phenotype. So these subunits come together as a tetramer to form their ion channels. The third class, and Stephen Tucker, who's here today, is going to talk more about K2P channels in humans and plants. So these have 4 transmembrane domains, but they have 2 pore loops per subunit. So these channels form as dimers, and they're thought traditionally to be leak or background channels, so you can see here through the IV curves. So the potassium channels that are expressed in fungi are structurally different. So they have 8 transmembrane domains and 2 pore loops per subunit. So again, it's not been proven directly, but it's thought that they form dimers. And functionally, they are intrinsically outwardly rectifying. So these channels, which are called TOC channels for 2-pore outwardly rectifying potassium channels, are found specifically in fungi, so you won't find them in animals, plants, insects and the like. So the thought is that these channels may make good antimicrobial targets for treating infections in humans, but also in plants where they get infected with agricultural pathogens. So what I want to talk to you about today are the basic characteristics of these TOC channels, because they're quite unusual. Propose some ideas for why they only pass outward current, and also begin to think about their physiological roles in yeast and whether they're regulated by environmental cues which govern morphological transitions in yeast. So we started by taking the sequence for the originally cloned the canonical channel from Saccharomyces SC TOC, which is here. Used it as bait to do a bioinformatics screen across all at the time, the sequenced fungal genomes, so there's about 205 genomes at the time. And we found 25 species of human pathogens expressed or encoded a TOC-like channel. We cloned TOC channels from Aspergillus fumigatus, Candida albicans, and then 2 strains of Cryptococcus neoformans. So this is variations neoformans and variation grubii. And when you express these in Xenopus oocytes, these are recordings through expression in oocytes by TVC, you can see that they generate robust currents, and from the IV curves here, you can see that they are sensitive to changes in extracellular potassium. So the black symbols represent 4K, the grey symbols 20K, and the white symbols at 100 millimole extracellular potassium. So they're all outwardly rectifying, and the threshold for activation follows the permeability profile. So generally they are potassium selective, but you can see here from these Nernstian plots that the Candida and Cryptococcus variants here do pass about 10% sodium, so they have a slightly shallower Nernstian profile. At the single channel level, the gating kinetics are quite flickery, quite difficult to analyse, but the NPO curve here fits well with an ohmic GV curve. So we really wanted to understand what was responsible for this outwardly rectifying properties. So even at very negative potentials, at very high external potassium, these channels don't pass any inward current. So the first thing we did was we thought that this was likely to be something to do with some kind of mechanism in the pores. So here's the alignments of the pore lining, transmembrane domain 6 and 8. So in grey here we have the selectivity sequences of both those pore loops. And we made a scan, mutagenesis scan of both of these transmembrane domains, mutating each amino acid in the Candida variant, Candida toc, to either aspartate, alanine, or asparagine. So I'm just going to show you the data from our aspartate scan. So down here you can see that there was 3 sites which generated inward currents when those residues were mutated to aspartate. So the first one is glycine 337, which corresponds to G99, the glycine 99 position in KCSA, which is known to be the gating hinge across potassium channels. And then the next one, V338, right next door, and then 5 residues down, another glycine which is conserved in top channels at position 342. So when you express, for example, the G342 aspartate mutant, you get currents that are rapidly activating, instantaneously closing, and in high extracellular potassium you start to see inward currents, and particularly in rubidium this is much more clear, and you start to see a linear IV relationship indicating that the channels are constitutively open. Another interesting point is that the channels become much more selective for potassium over sodium when this mutation is made. And this is confirmed as well through the regulation by, or the block by barium. So the mutation is less sensitive to barium block. So coincidentally, it turns out that the residues that we hit had been described before. So, as I said, the glycine in KCSA at position 99 is known as the gating hinge, and 5 residues C-terminal to that is in MTHK is an alanine, and this is the narrowest part of the pore in MTHK. And the equivalent residue in herd channels, which is also an alanine, when you mutate this residue, it forms a constitutively open channel, and that's equivalent to this glycine here at 342 in Candida, but also across all the other top channels. This is also true in mammalian K2P channels and other K2P channels. The equivalent glycine in Kc and K0, which is the Drosophila K2P channel, when mutated to an aspartate, also confers constitutive activation as well. So the mechanisms that allow for the channel to outwardly rectify, the components are already present in all the other channels, so they're not completely different from other potassium channels in that respect. Just for completeness, we can make mutations at the other site. So this is the V338 mutation to aspartate, again, rubidium gives a much clearer picture of constitutive activation. And this occurs also if you make the same mutations across other members of the family at the same positions, you also get a break in the rectification. So then we wanted to consider how we might explain this. So we came up with a rather simple model that we're trying to now challenge. So you should know that, I mean, generally there are 4 binding sites for potassium inside the pore, 1, 2, 3 and 4, and then there's an external and an internal potassium binding site as well. There are different models that describe how potassium permeates the pore, but the canonical model infers that potassium is either bound at the 1st and 3rd sites, and this interchanges with binding at the 2nd and 4th site. So what we proposed is below EK or below EREV, where the driving force is inwards, the potassium would occupy the S1 site and S3 site, leaving the S2 site vacant, and this has been shown in KCSA to induce rapid inactivation and pore collapse, which might explain why we don't see any inward current. Conversely, when the driving force is outwards, so above EK or above EREV, we see, or we propose that there's more occupation at the S4 site, and therefore S2 is now occupied and the channel is conductive. There are other facets of the behaviour of top channels that fit with this as well. So we see in high potassium that the current collapses. So you might have noticed on the IB curves at the beginning that the current collapses at high K, more than would be expected if it was just due to a change in driving force. And we suspect that this is because at high K we get more occupation of S1, and so the pore becomes non-conductive, and so you get less current in high K conditions. So I just wanted to now go on and think about roles of top channels in fungus. So again, focusing on the Candida variant. So Candida albicans specifically is the leading fungal infection in the immunosuppressed population, particularly those who are undergoing treatment for HIV. Candida exists in multiple morphological states, either as a ball, a yeast form, or in more extreme circumstances, they can filament and generate these hyphal finger-like projections which help to increase its virulence properties. The cues that dictate whether the yeast is in the yeast form or the hyphal form range from body temperature, for example, pH, fungal pheromones such as farnesol, which is excreted by Candida albicans itself. And for example, in acidic pH, the yeast likes to reside more in this form, and then when you change the pH to more alkaline or neutral, it tends to induce hyphal formation. So the question was, are top channels involved in controlling any of these morphological transitions? So I'm not going to show you any microbiology today, so I'm just going to show you the sort of the evidence that from the biophysics. So there's indirect evidence previously that when you activate top channels, so this is Saccharomyces top, can be activated by the anaesthetic isoflurane, and isoflurane actually coincidentally prevents hyphal formation. As I said on the last slide, there are a number of other types of cues which dictate morphological transitions, so low pH, farnesol, and arachidonic acid. When you express Candida toc in oocytes and you add farnesol, it causes an increase in current, the same with arachidonic acid as well. So it suggests that activating these channels may well, at least coincidentally, may suggest that these channels may be involved in regulating morphological transitions. In terms of pH sensitivity, We've looked at the pH sensitivity of the channels because the mechanism which dictates the transitions in Candida albicans are not known yet. The actual pH sensor has not been identified, although the signalling pathway is well described. So we looked at pH, and as you reduce the pH to more acidic values, you see an increase in the Candida albicans top current, and not in the Saccharomyces variant, which is pH insensitive, certainly to extracellular pH. Interestingly, as you decrease the pH to more acidic values, you see a shift in the reversal potential towards EK, so the channels become more selective. And to cut a long story short, we identified a histidine residue here between S1 and S2 in the S1, S2 loop, a histidine in position 144. When you neutralise this, it largely removes the pH dependence of the channel. You can make unphysiological mutations, so if you change the histidine to a lysine or to an aspartate, you can recapitulate the effects of changing pH on the channel. So with the lysine, obviously it's constitutively protonated, so you don't see any pH response. It's highly selective for potassium, whereas the aspartate mutant is not. And in fact, the lysine tends to reduce sodium permeability from about 10% down to about 3%. In terms of what is happening in terms of the increase in the current, so if you look at single channels, using these mutations, the lysine and the aspartate, it doesn't appear that single channel conductance changes, but in fact it is the open probability that changes. So the increase in current is due to a change, at least we think, in open probability. So this residue sits all the way over here. I know this is only a 2D picture, but it sits all the way over here. We don't know how the channel is folded because there's no structural information. But we wanted to know if this histidine may well talk to any other residues around the pore. I'm not going to show you the data for, we looked at a lot of non-conserved charged residues that are exposed around the pore areas, and they don't seem to be involved in conferring pH sensitivity. But what we did do is we did a bit of a mutagenesis scan again, so we split the two pores into three parts, and we replaced the Candida amino acids with the equivalent Saccharomyces amino acids, so from the non-sensitive channel. And just to save time, we did this in groups. And when you test the pH sensitivity by changing the pH from 7.6 to 5.6, you can see that even with an almost entirely Saccharomyces P1, there was no effect on pH sensitivity. But there does appear to be something that's required at the distal end of the second pore subunit here. And when you mutate individually each of the different residues from the Candida to the Saccharomyces, you find that there is one residue, a serine at position 466, which is right here at the top of TM8, which is critical for allowing pH sensitivity. If you mutate this residue to anything else, so to isoleucine or threonine or cysteine, there's no pH sensitivity at all. So it's a serine that has to be required in this position. We're not quite sure if it's the histidine that talks to the serine, or whether it's just structurally a serine at the positions required for the changes that are induced by pH in the pore. So I've shown you that fungi express a structurally and functionally unique potassium channel, which is not present in any other living organism. And so that makes it an exciting target for future antimicrobial research. These channels are a curiosity. They are intrinsically outwardly rectifying, and we do provide a rather simplistic model, but it does seem to fit with all of our data to explain how the rectification process might occur. There are a number of chemical cues which promote morphological transitions, and all of these that we've studied so far seem to activate top channels, which may indicate that these channels are components of the morphological transition process. And specifically, we've gone into some detail looking at pH regulation of these channels, and we know that this is dictated by an extracellular histidine in the S1-S2 loop, but also that there's a serine residue at the proximal end of S8, the last transmembrane domain, that sits behind the second pore loop, which is required to confer these changes. So, just like to thank a number of people who've worked in my lab and collaborator, Andy Corran at Syngenta, and also Luis Coelho and Steve Goldstein in the States. Thanks very much.
Thank you very much, Tony.
Thanks, Tony.
Do you have any questions for Tony? Damien, do you want to start us off?
Lovely data again, Tony. Very detailed studies. It's interesting, and you may have some data on this, but you may be keeping it back. Have you done any pharmacology on these? Because, I mean, obviously that similarity with the other two-pore potassium channel, particularly in the pore lining residues,
Yeah.
that TVGYG or TIGYG, you might expect, for instance, pore blocking two-pore potassium channel modulators to potentially act on it. So I don't know whether there's some hints towards where some of that kind of pharmacology may be leading.
Yeah, so we know that certainly the Candida variant is sensitive to TEA block in a voltage-dependent manner. And this is interesting because one chemical, lactoferrin, causes apoptosis of Candida albicans, and it does this through releasing potassium from the Candida. And you can block that process with TEA. And that was in a paper a few years back, not from our lab. But we know that the channels are TEA sensitive, which might explain that. They seem to be barium sensitive, apart from the Aspergillus variant, which is less so. We went through a whole host of activators, and pretty much they were insensitive to most of the potassium channel activators. Some of the K2P channels, mammalian K2P channels, are sensitive to BL1249. And this is actually a variant from the plant pathogen Septoria. It's also sensitive to BL1249, as is Candida. And other phenomates as well are also activators, but phenomates seem to activate pretty much most stuff. So, but at least it shows that they're not completely different, which I'm not sure is a good thing or a bad thing. But yeah.
Gives you a start point, and then it's trying to diversify away from that start point, I guess. Thank you.
Any other questions?
On a related point, what's the therapeutic strategy to inhibit the channels? That's what you
No, to activate them. Well, yeah, I guess it's a bit confusing. So if you activate them, then you're more likely to, well, we propose you're more likely to reside in the yeast form, which is less virulent. But also, my former mentor had a study looking in Saccharomyces where you could activate the channels perpetually with the toxin, and this ended up causing potassium dyshomeostasis, and the yeast just die. So activating the channels seems to be the way to go, but it's obviously more difficult to design efficient activators than it is to block them. So yeah.
Any other questions? Yes, at the back, please.
Many thanks for the exciting work. it's more like a comment. As you said, because of the highly flickering nature of the gating, it's quite challenging to do kinetics. And I presume also to reliably measure the unitary conductance as well, because of a lot of missing events and other things. So I believe that you tried your best to use as high sampling rate as practicable to do the single channel recording, right?
Yeah.
Okay.
Yeah. My former boss did quite an in-depth study of the single channel. Yeah, so they're actually not open very much, but when they do open, they tend to sort of flicker quite a lot. And the other interesting thing is that there's actually not many of these channels in a yeast. So someone managed to predict that there's probably only about 50 ion channels in an entire yeast. So again, that kind of complicates.
The other comment or question I have, like these channels, based on what I've seen, they tend to manifest a lot of multiple opening.
Mm-hmm.
And some people say it's different channels, some like multiple channels opening. Some people say it's a double barrel gating. So any of your mutant, did you see any change in this multi-channel behavior? Or
We haven't looked at the mutation in too much detail. Certainly the glycine mutations, we have a little bit of data, but they are particularly difficult to record. So yeah.
many thanks. Thank you.
Great, I think we'll finish there. Thank you very much indeed, Tony.